Hi everyone, this is Lecture 9, Neurophysiology Part 2. So we are going to continue where we left off with neurophysiology. The last lecture, we introduced you to the anatomy of the neuron, which is the primary signaling cell in the nervous system, and we talked all about electrical communication in neurons, how graded potentials and action potentials activate neurons, and what those signals look like and how we measure them with membrane potential. So today we're going to do chemical communication in neurons, which is what happens after the action potential reaches the axon terminal. We'll talk about neurotransmitters. We're going to talk about also how to increase signaling in neurons through nervous system pathways. So neurons communicate electrically through electrical gradients. Those are the sodium and potassium gradients that we talked about in the previous lecture, where the movement of ions in and out of the membrane is the electrical signal that travels through the cell. They can either be small and local in the case of graded potentials, or they can travel long distances down the axon of the cell, and those would be action potentials. Once the action potential reaches the end of the axon or the axon terminal, there will be chemical communication or release of neurotransmitters. A chemical is also referred to as a ligand, so you may want to go back and review your cell communication lecture where we talk about ligand receptor interactions. So the release of neurotransmitters is received in the next neuron by neurotransmitter receptors. So as a reminder, here's the anatomy of the neuron. So the action potentials are going to travel down the axon to the axon terminal. And the axon terminals will contact the next cell at the synapse. So a synapse is a connection point between two neurons, or between a neuron and a muscle, or between a neuron and a gland. So the most common will be synapses with other neurons. Those we just call general synapses. And here they are. So action potential will go all the way down the axon, and then at the axon terminal, we will have the contact with the next cell. So here in purple is the second neuron, and these are the dendrites, where the input from the previous cell will be received. So the axon terminal is going to synapse or connect to the dendrites, sometimes the cell body, of the next cell. In the case of a neuromuscular synapse or a neuromuscular junction, this is a neuron where the action potential travels down the axon, and at the end of the axon terminal, they will synapse with skeletal muscle cells. They will then send input to the skeletal muscle cell so that the muscle knows to contract. We'll talk more about this in a couple of lectures when we talk about muscle physiology. The last case is the neuroglandular synapse, where a neuron can send action potentials down its axon to contact glandular cells. And in this case, activation of a glandular cell will lead to secretion of chemicals. So we're going to complete our neuron communication concept by talking about the electrical to chemical communication flow. So at the connection between two neurons, there is a space. The space is called the synaptic cleft. The synaptic cleft does not allow electrical current to flow. So we can't just stay electrical, like we do the wires in your house, and just connect electrical to electrical. We need an intermediary that can cross the synapse. And the intermediary is the neurotransmitter, which is a special chemical released at a synapse that will signal to the next cell. That chemical signal will then be received, and the next cell will have an electrical potential generated. That can either be a small signal, which will be a local graded potential, or it can be a large signal if there's enough neurotransmitter release and will turn into an action potential that will activate completely the second cell and further the chain of communication. So let's talk about the structures of a synapse, and we are going to draw this out. So let's draw it out. Okay, so at a synapse, what you have is the axon, which will carry the action potential, traveling in, and then it's going to end in this little bump we call the axon terminal. So, whoops, the axon is going to come down and end in the axon terminal, and that is what is going to carry the action potential. I'm just going to abbreviate it as AP. So the action potential is going to travel down the axon. We have the axon terminal, and then we have a space. And this space is called the synaptic cleft. Then it will contact the dendrite, or the muscle, or the cell body of the next cell. This we call the postsynaptic cell, and the cell that has the axon with the axon terminal is the presynaptic cell. Located in the axon terminal are vesicles, which are kind of like tiny little bubbles that are filled with neurotransmitter, and these we call synaptic vesicles. Located on the postsynaptic cell membrane, which will be the dendrite or the muscle of another cell, are going to be receptors, and they're specific to the chemicals that are released by a neuron. So these are neurotransmitter receptors. Okay, so let's look at the pictures from the textbook. So we have the presynaptic neuron with its axon coming down. There's a myelinated axon in this case, so that's myelin wrapped around the axon, with the synapse here onto the next cell. And in this example, there are 3 cells that this particular axon is connecting to. And then we zoom in just on this one single synapse, and you'll see here we have the axon terminal with the axon coming in, the synaptic cleft, which is the space between, and the postsynaptic cell membrane, which is usually the dendrite. So within the presynaptic neuron, we're going to have synaptic vesicles. These are tiny little sacs or little bags of chemicals that will be released when the signal reaches the axon terminal. Within the synaptic vesicles are the chemicals that are going to excite or send signals to the postsynaptic cell, muscle, or gland, and those are the neurotransmitters. The synaptic cleft is the space between, and remember that electrical signals cannot pass through the synaptic cleft. That's why we need to have neurotransmitter released here. So the chemicals are able to pass over the synaptic cleft. They'll be released, they'll pass over the synaptic cleft, and they will be received in the postsynaptic neuron, the postsynaptic membrane, at the neurotransmitter receptors. These are proteins, just like any other receptor we have learned about, that are found in the postsynaptic cell membrane. They will specifically bind chemical neurotransmitters, and they will receive that signal. The neurotransmitter receptors often are ion channels, or they are coupled next to and very close to ion channels. So once the neurotransmitter is received by the receptor, an ion channel will open. And if you remember the last lecture, what happens when ion channels open? we get movement of charged ions. So when an ion channel opens, we can either excite or otherwise make electrical changes in the next cell. So here's a close-up of a synapse, probably a bit better than the one that I drew, but there's your synaptic vesicles in your axon terminal. There's your neurotransmitter being released. These are the protein receptors that are the neurotransmitter receptors that will specifically bind the neurotransmitter. This particular one is also an ion channel, so when it opens, ions will flow through and you will get electrical changes again in the postsynaptic membrane. So we've gone from electrical through the axon transmission of the action potential to chemical through the chemical release of neurotransmitter, and then back to electrical where the ions will move into the postsynaptic membrane and make changes in the postsynaptic cell. Okay, let's get a little more formal here and talk about the specific events at the synapse. And we're going to break it down into 6 steps, 3 in the presynaptic axon terminal and 3 in the postsynaptic axon terminal. Okay, so first the action potential is going to reach the axon terminal. It's going to travel down the axon terminal, and this will depend solely on electrical conduction. Do you remember the 2 things from the previous lecture that affect electrical conduction? Think about how you would want to get the fastest conduction. What kind of an axon would you want to be the fastest possible? The fastest conduction will be a myelinated large axon. The slowest conduction will be a non-myelinated small diameter axon. So this action potential reaching the axon terminal is going to depend solely on electrical conduction through that axon. That is, of course, assuming that the action potential was received and reached threshold. So, of course, there are graded potentials. Anyway, we won't get into that. So this is through electrical conduction down the axon. That's step 1. The action potential is good down the axon terminal. The action potential actually opens up here in the axon terminal some voltage-gated calcium channels. Now we've talked about voltage-gated sodium and potassium channels, which help us with the action potential. Here's another type of voltage-gated channel that does not cause an action potential, but what this causes is release of neurotransmitter. So these voltage-gated calcium channels are waiting for the action potential to come in. That change in voltage that is caused by the action potential will open up these voltage-gated calcium channels. Calcium will come in and the neurotransmitter will be released from the synaptic vesicles. So the synaptic vesicles are sensitive to calcium changes and they will know when calcium has entered and they will dump neurotransmitter into the synaptic cleft. So that is step 2. Calcium enters the axon terminal. Step 3 then will be the actual neurotransmitter release that is caused by the calcium. So calcium entry into the axon terminal triggers exocytosis or release of the neurotransmitter into the synaptic cleft. Step 4 is that the neurotransmitter is binding or received by the neurotransmitter receptor. So here in green are these proteins in the membrane of the postsynaptic cell. They are going to bind to and recognize the neurotransmitter has been released. And that will then lead to step 5, which is the ion channels opening on the postsynaptic cell. So the neurotransmitter binding to the receptor causes the opening of ion channels attached to this receptor. Ions will then move. The ions aren't pictured here, just the neurotransmitter, but the arrows show that the ions now can move through this opened channel. So what type of channel are these ion channels? How are they gated? Think back to our cell communication lecture. We talked about several types of gating channels. These are ligand gated or chemically gated channels. So the neurotransmitter binding is what opens the gate on these neurotransmitter receptor channels. The last step is that influx of ions, or the change in ions in the postsynaptic cell membrane, causes a graded potential. Why doesn't it cause an action potential? We seem to be very focused on action potentials, and it is a complicated process, but that's not actually how it always happens. These are small. This is a single tiny axon terminal in one part of the input to the next cell. So this little bit of neurotransmitter typically will only cause a small graded potential at the dendrite. So just a little bit of change. That's going to be a small local change in ion flow in the postsynaptic dendrite. So if we summarize these events, let's draw them out and make sure you guys understand what's happening, okay? Okay, so we are actually going to just go back to our previous drawing, so we don't have to draw everything again, and I'm just going to use another color to show you guys the different steps. Okay, so step 1 is action potential. So step 1 is that we have an action potential traveling down into the axon terminal. Step 2 is going to be calcium entry into the axon terminal. And how does that happen? That happens through voltage-gated calcium channels that are located in the axon terminal. So calcium is going to come into the axon terminal as a result of the voltage changes from the action potential. Step 3 is that this calcium is going to trigger the vesicles to release neurotransmitter. So let's write that over here. Step 3 is going to be neurotransmitter release. Because of that calcium signal, the neurotransmitter will be released, and then step 4 is going to be the binding of the neurotransmitter and the activation of the neurotransmitter receptor. So step 4, activation or activate. I'm just going to abbreviate them NT receptors. Again, this is a ligand receptor interaction. So the calcium entry is voltage-gated calcium channels. The neurotransmitter receptor activation is ligand or chemically gated channels. And it turns out in this example that these ligand chemically gated channels are attached to ion channels, and they are going to then cause step 5, which is ion movement, or ions enter the postsynaptic membrane. Then the final step is that this ion change causes a graded potential. So if we had an electrode inside this postsynaptic cell, we would measure a change in voltage as a result of the neurotransmitter activating the receptors and the ions entering this postsynaptic cell. So this is a summary of the events at a synapse. And let's go just a little bit more into what happens with these graded potentials. So you will notice I didn't tell you what ions enter the postsynaptic cell. That's because it depends on the type of postsynaptic cell and the type of neurotransmitter that is released. So the most common is that the neurotransmitter will excite the postsynaptic cell. So if that graded potential is excitatory, meaning that it moves in the depolarizing direction up towards zero, then we call that an EPSP. And how do we depolarize a membrane? Think back to our ion changes that we talked about in the last lecture. Most commonly, depolarization is caused by sodium entry. So, if sodium enters a synaptic membrane, then this will be an EPSP or an excitatory graded potential. This is used to increase depolarization. This can get the cell closer to threshold, and it can build up to lead to action potentials. There is another possibility, which is also important for balancing the nervous system. That is that negative ions can flow into the postsynaptic membrane. For example, this often happens with chloride channels. If a negative ion flows into the postsynaptic membrane, meaning that chloride channels are opened by the neurotransmitter, then this will be inhibitory. This is going to hyperpolarize or bring the membrane potential down further from rest, and this will get us further from threshold. That will overall make it more difficult to get an action potential, and an IPSP can quiet a cell or reduce the number of action potentials produced. If you guys know anyone who has epilepsy or similar type clinical conditions, this balance of excitation and inhibition is very important in the nervous system. Often, individuals with epilepsy have some imbalance in the excitation inhibition such that regions of their brain are overly excited, and that can lead to seizures. So a lot of epileptic medications will actually increase the IPSPs or the inhibitory neurotransmitters to try to balance out that overstimulation that's happening in the epileptic condition. So whether we get an EPSP or IPSP, whether we get excitatory or inhibitory effects based on the synaptic transmission, is going to depend on the type of ion channel that's activated by the neurotransmitter. And that will depend both on the neurotransmitter and the neurotransmitter receptor. So here graphically is how we can look at these graded potentials that happen after activation of the synapse. So remember that our resting membrane potential is around -70, and we will get an action potential that comes in from the presynaptic cell, and then we're recording here from the postsynaptic cell. So we get the neurotransmitter release, the neurotransmitter comes in, activates the ion channels, sodium moves in, and we get a small EPSP, usually around 5 millivolts or so. And that gets us closer to the threshold that could lead to an action potential. The opposite would be if we have a neurotransmitter and receptor pair that causes an increase in chloride or negative ions into the cell, and that will take us from rest below rest or hyperpolarizing, and that will give us an IPSP, and now we're even further from threshold than we were at rest, and it will be even more difficult with an IPSP to activate and get to threshold. So reaching threshold depends on many inputs to one cell. EPSPs and IPSPs will enter from multiple synapses, and it's really a combination of activity that is going to be read by the cell. So we'll start at resting potential -70 millivolts, and there are many ways, many combinations that could get us to threshold, which is -50 to -55. So graded potentials plus or minus 10 millivolts millivolts, probably an exaggeration here, but this is kind of where your textbook puts it. You know, they can be 1 millivolt, 5 millivolts, 10 millivolts, okay. Graded potentials will add up to get us to threshold. There are two ways that graded potentials can add up, and this is called summation. So a single EPSP alone cannot induce an action potential, but they can be added or summed. The axon hillock will keep score of all the graded potentials until we get to threshold. So here's all the inputs at the dendrite and cell body of this postsynaptic cell, and the axon hillock here is adding them up, adding up the EPSPs, subtracting the IPSPs, and hoping we can get to threshold. If threshold is reached, then we'll have an action potential. So two ways. First, we can have temporal summation. Second, we can have spatial summation. Temporal summation is when you have one presynaptic neuron. So here's our single presynaptic neuron. It's an excitatory neuron, which is abbreviated here as E1, releasing excitatory neurotransmitter. One single graded potential from this excitatory cell is not enough to reach threshold. It's just below threshold. You would need more than one to build up. So in temporal summation, we're going to take this same location, and instead of just having one EPSP or one graded potential, we're going to do two really close together in time so that they add up. So here's 1, 2, really fast. And these are fast, which means we increase the frequency, so we're going to go dot from the same cell. When we get two graded potentials very close together like that, they can build up, reach threshold, and then look at this. Do you guys recognize that shape? There's an action potential. That is an all or nothing event that happened because the graded potentials added up. The next way to add up graded potentials is spatially, and this is by using multiple inputs. So here's E1, or excitatory cell 1, here's E2, or excitatory cell 2. And in this case, rather than going dot, or having them very close together in time, we're going to keep the same timing in E1, and we're going to add a second cell that will fire or send a graded potential together. So we then can add up those two together in space. So for frequency, we keep the same space and we increase the timing. For spatial, we're going to keep the same timing and we're going to increase the number of cells or the locations that are being stimulated. So in this case then, E1 plus E2 will also get us to threshold, and then again, action potential. Okay. If instead of adding an excitatory graded potential to E1, we add an I, or an inhibitory graded potential, that inhibitory graded potential can cancel out the excitatory potential from E1. So in that case, even though we had two neurons active in space at the same time, one of them was inhibitory and it canceled out the excitation from the other one. So neurons are always adding together their inputs, taking in all the information from everybody that is talking to them and deciding whether to continue on the signal. So if we have two excitatory inputs that are too slow, they can't add up in time. But if we put two excitatory inputs very close together in time, then they will get an action potential. Or we can take two separate cells and we add them up in space very close together, then we can get an action potential. If they're too far apart, the graded potentials are local, so they will dissipate. That won't work either. So they have to be close together in space. They add up, action potential. We cannot get a threshold from adding an inhibitory input. That's going to quiet the cell or cancel out the excitatory input. So this is all done through neurotransmitters. There's many types of neurotransmitters in the nervous system. In general, one neuron type releases one type of neurotransmitter, but any given neuron has multiple receptors. So any given neuron can respond to both inhibitory and excitatory neurotransmitters. So usually we talk about acetylcholine. This is the most common one that's found at neuromuscular junctions. It's very common in the autonomic nervous system when we talk about the fight or flight and rest and digest nervous systems. But we also have several other neurotransmitters in the brain. So we have glutamate, which is the main excitatory neurotransmitter in the brain. We have GABA, which is the main inhibitory neurotransmitter in the brain. We have norepinephrine, which is related to adrenaline, and that is found in autonomic nervous system, learning, emotion, behavioral pathways. We have dopamine and serotonin, which are also very common in learning, emotion, and behavioral pathways. So we can classify neurotransmitters by their chemical structure, and why would we do this? Why is this important? Well, think back to your cell communication lecture. How do chemicals or ligands affect receptors and cells? It depends on whether they are water soluble or lipid soluble. So classifying neurotransmitters by their chemical structure helps us understand a little bit more about the types of ligand-receptor interactions they may have. Now, we could spend an entire class doing this, but I'm just going to give you guys some of the categories. So acetylcholine, kind of on its own. Biogenic amines will include the catecholamines and serotonin. Amino acid-based neurotransmitters will be glutamate. There's also glycine and GABA. Peptide-based will be endorphins and substance P. Chemical messengers can also act as neurotransmitters, and those can be ATP and nitric oxide. When these neurotransmitters become important for different pathways, we will let you know as we're studying various pathways throughout the rest of the course. So we can also classify neurotransmitters by function. So we can have excitatory neurotransmitters that will cause EPSPs or depolarization, like glutamate. We can have inhibitory neurotransmitters that cause IPSPs or hyperpolarization. Examples of that, the most common is GABA. Or some neurotransmitters can do both. And they do that by acting at different receptor types. Acetylcholine is the most common. Acetylcholine is always excitatory at a muscle because it acts on a particular type of receptor that's called a nicotinic receptor. But it can also be inhibitory. For example, acetylcholine, when it reaches cardiac muscle, interacts not with the nicotinic receptor but with the muscarinic receptor, and that is an inhibitory receptor that will lead to IPSPs in the cardiac muscle. More on that when we get to those specific pathways. How does this happen? Neurotransmitter receptor mechanisms are just like the ligand receptor interactions we looked at in our cell communication. So this should be somewhat review for you guys. They can either be direct, where the ligand binding to the receptor or the neurotransmitter receptor interaction directly opens ion channels, and then ions will flow into the cell. Examples of these will be acetylcholine and amino acid-based neurotransmitters. These will be very fast interactions, directly opening the channel, ions go in immediately. The indirect way is through second messengers. So go back to your cell communication lecture and look up second messengers. This is where you have a cascade of protein and enzyme changes that are caused by the ligand receptor interaction. So here's the neurotransmitter binding to the receptor. That receptor then will be coupled to a G protein, which will trigger a series of domino events within the cell that can then cause changes inside the cell. It doesn't mean that it's not going to cause ion changes. It can and absolutely will open ion channels, but it can also do other things like affect enzyme activation and protein synthesis. So examples of these would be biogenic amines, peptides, and some of the dissolved gases like nitric oxide. These will be slower effects, although they're still fast for the nervous system. They're not slow like hormones, and they will be long-lasting. They will last a bit longer than other neurotransmitter effects. So neurotransmitter receptors will mediate changes in membrane potential according to the amount of neurotransmitter that's released, how long the ligand binds to the receptor, and the amount of time that the neurotransmitter is bound to the receptor. So this will be the dissociation or the release of the neurotransmitter. So more neurotransmitter coming in is more receptor interaction. And then eventually, just like all chemicals do when they bind, they will unbind the receptor, and that will turn off the signal. So neurotransmitters will affect the membrane potential as long as they are bound. So they can also be deactivated. So there are 3 different ways to stop the neurotransmitter signal. Graded potentials will continue as long as the neurotransmitter is bound. So we have to inactivate or remove it in order to stop the signal. So there's 3 ways. One is active transport. One, we can pump the neurotransmitter back into the presynaptic cell or into the nearby glial cells. The second, sometimes there are enzymes in the synaptic cleft that will break down the neurotransmitter so that it no longer activates the receptor. One example of this is acetylcholine esterase, which I'll show you in a minute. The second is diffusion. Sometimes the neurotransmitter will diffuse enough away from the synaptic cleft that it will no longer bind to the receptors, and that will reduce the signal. So these are visuals of those 3 different possibilities. So here's your synaptic vesicle with a neurotransmitter being released. As long as it's bound to the receptor, you will have changes in the postsynaptic cell. So to get rid of the neurotransmitter, we can pump it into nearby glial cells, which will recycle it. We can break it down with enzymes so it no longer binds to the receptor, or we can diffuse it away through the extracellular fluid, through the local bloodstream, and remove it from the terminal that way. One of the important ways to do this is through enzymes. So acetylcholine esterase is a common enzyme found in the synaptic cleft that breaks down or degrades acetylcholine. So here's the reaction. Acetylcholine will break down to acetate and choline with acetylcholine esterase present. It can then actively be transported back into the presynaptic terminal and recycled to reform acetylcholine in the synaptic vesicles. That uses another enzyme called CHAT, or choline acetyltransferase, which will combine choline with acetyl-CoA to make acetylcholine back in the synaptic terminal. So there is constant recycling of the neurotransmitters to stop the signal and then to reform and reset the synaptic vesicles. This is a visual of the enzymes involved in the acetylcholine recycling. So here's acetyl-CoA and choline being combined to make acetylcholine and put into the synaptic vesicles. The acetylcholine will be released and bind to its receptor, but over time acetylcholine esterase will degrade the acetate and the choline, and then that can then return to the synaptic terminal where it will be recycled. The detail here of acetyl-CoA coming from the mitochondrion should be familiar if you remember what mitochondria do for metabolism, and that is one use of extra acetyl-CoA. The reason we need to think about stopping signals is because there are many clinical conditions that are caused by overactive or underactive synapses. And so from that, we have many drugs to treat clinical conditions that can alter synaptic activity. There are many drugs that change the synthesis, storage, or release of neurotransmitter. There are drugs that can change the interaction of the neurotransmitter with the receptor, make it more or less. There are drugs that can influence the reuptake or destruction of neurotransmitter to make neurotransmitter reuptake last longer or make it stronger. And we can also have drugs that replace deficient neurotransmitter based on some clinical conditions with a substitute neurotransmitter. So 2 examples, serotonin reuptake inhibitors, or SSRIs. My husband calls these happy drugs, right? Prozac, related other medications for people that are treated with depression and other conditions. Often, what is believed is that they do not have enough of this particular neurotransmitter in their system. So they will prevent the reuptake of serotonin, which increases the length of time that serotonin is available. That will increase activation in an individual that does not have enough activation of these serotonin synapses. In other words, they're depressed. So increasing that activation gets more serotonin released, more active synapses, and that can help patients who are suffering from clinical depression. Another example is tetanus toxin, which prevents the release of GABA from inhibitory neurons at the neuromuscular junction, and this can cause uncontrolled muscle spasms. Too much excitation, locking up of the muscles because they're not able to release, and that is why tetanus is often referred to as lockjaw, because you'll lock the muscles in your jaw and they will get stuck that way because you can't unlock them. Okay, so neurons are going to communicate electrically and chemically. The electrical communication happens through the movement of ions in and out of the cell membrane. These can be graded potentials, which can be small, local, EPSPs, IPSPs, or long-distance action potentials. If the EPSPs and IPSPs add up enough to get to threshold, then we'll get an action potential that will travel a long distance down the branch of the cell. Once the action potential reaches the end of the cell, or the axon terminal, there will be release of neurotransmitter. That's going to depend completely on that type of cell, what neurotransmitter is released. The release of that neurotransmitter will interact with the neurotransmitter receptor on the next cell, or the postsynaptic cell, and it will have an effect on that cell, which will either be EPSPs or IPSPs, and that will depend on the receptor ion channel interaction in that postsynaptic cell. So overall, the nervous system is organized into very complex pathways of multiple cellular connections. Understanding the nervous system requires an understanding of how groups of multiple cells interact as neural circuits. So you can already see just in the adding up of the EPSPs and IPSPs, that's only one cell, right, with all of its inputs. Now we're going to get to 100 billion neurons in the nervous system with over 10 to the 14 synapses. So now think about all of the EPSPs, IPSPs that are adding up all the time, all of the action potentials that can happen, and all of the signaling. It's kind of mind-blowing, right? So any given neuron can be connected to 5,000 to 10,000 other neurons. Can be. It's not always the case. Sometimes it's 1-to-1, but sometimes it's in the thousands. So when people say that your brain is like a computer, I think that is actually a serious oversimplification. Modern computers cannot come close to the recognition, processing, and learning that our nervous system is capable of. It is a fascinating problem, though, for computer scientists to play with. So to think about pathways, there are 2 ways to think about the input and output of the cell. So a single cell is influenced by thousands of other cells. Look at all of these cells coming in as inputs to that one cell. That's called a convergent pathway because all of those cells are going to converge on one single cell, and then that cell will add up those inputs and decide whether to be active or not. From that cell, then, we can also branch out to thousands of other cells. And when we branch to thousands of other cells, that is diverging, or if that cell decides to become active, we can now send input diverging out and affect many other cells. So divergence is the potential output of a neuron. So information coding in neural circuits is really fascinating. If you're someone who loves to think about computer science and the basic mathematical calculations that can go with all this, this can get really fun. But the bottom line of this is that ultimately the information conveyed within a neural circuit is found within the patterns of electrical stimulation, and recording from the brain is absolutely fascinating to listen to the of all the action potentials that are going on. So the action potential patterns are affected by the graded potential patterns. So graded potential patterns will be, remember they are small, subthreshold EPSPs and IPSPs. We can increase the amplitude and frequency of those inputs based on more or less activation. So what we're going to talk about in the next lecture is sensory communication and activation of receptors. And activation of sensory receptors can change the amount of graded potential amplitude size or the timing of graded potentials coming in to a cell. If we get enough graded potentials, then they will add up to an action potential. But remember that you cannot change the shape or the amplitude of the action potential. The action potential always looks the same. So the only way to change an action potential pattern is to increase the timing of how often an action potential occurs. So it is the da versus da that is used as the coding signal, kind of like the Morse code of the brain. And the frequency or the change in pattern of action potentials is how your brain then transmits different information throughout different circuits. We could talk about this forever. It's super interesting, but I think we've had enough for today. So finish this up. Please redraw the synapse if you guys need to spend a little bit more time thinking about the events at a synapse, and let me know if you have any questions.